Okay, so we want to do a quick review of the gene expression module. Let's just start from the beginning of the first lecture. We talked about next generation sequencing. We are now sequencing hundreds of thousands of hundreds of millions of reads in one lane. So for example, there are CRO companies now. We can get about 400, 500 million reads for $1,200. It's unbelievable. And each paired end read fragment is PE 150. PE 150 times 400 million for only $1,200. It's really quite amazing. And for quality control, we talk about FastQC. And then for just raw read mapping to the human genome, we talked about using Boros BW transformation and also LF mapping. There are two different algorithms, BWA and Bowtie, and both are quite widely used. And after you map the FastQ reads into the genome, you will get either a SAM file or the binary compressor file, or you can also get the BET file. Although people use BET file mostly for intervals, right? For raw mapping, usually they use SAM or BAM. And there are also tools like SAM tools or BET tools which can manipulate SAM files and BET files. And then we just very quick, by the way, we will be discussing more of this part in the next second module. That's when you really use BWA. Very quickly, we went to RNA sequencing. And so for that, the read alignment needs to be more splice aware. We focus on just mapping the exons and the junctions, right? So there's STAR, there's HiSET, and also there are de novo alignment tools, which, for example, for cufflinks, it does not need to even have a known transcript annotation. It just starts trying to map to the genome and figure out where the exons are and where the junctions are. Trinity is a different tool. By the way, we didn't really talk as much about this. We focus mostly on STAR, and that's what you are using for homework. Both HiSET and STAR are used quite a bit. For the de novo, I just want to quickly mention that. So cufflinks is if you don't even know any transcript ahead of time, you can use it. And Trinity is even more powerful in that supposedly if there is an alien that they still use DNA, you don't even know the genome sequence for this species, you can still do the RNA expression as long as they also use DNA and RNA too for encoding their genome. Yes, question. No. So STAR and HiSET, you can give it a transcript annotation, which will speed up a lot of things. It will still try to create some junctions, but it can start to use your known exons a lot. Cufflink does not need that, and Trinity doesn't even need to know the reference genome. So that's pretty cool. For quantification, so we mentioned RSAM. This is a tool that started really looking at transcription level using TPM. So they were the first method to propose using TPM to represent a more accurate way to represent gene expression for RNA-seq. You can still get the normalized count, and you can also use FPKM or RPKM. We also talk about pseudo-aligners. So these are Callisto or Salmon. Both can be run directly on the FastQ file. They can also be run on a BAM file, say coming from STAR, which will be a little faster, but it will also give you a TPM very quickly. The pseudo-aligners only care about the known transcript expression. And then we talked about differential gene expression. And so this is actually calculated at the normalized read counts. So you want to keep the read count because the long genes, you do have more reads, you will have better statistical power to detect the differential expression. And we mentioned that the number of reads that really land on a gene in different samples, they follow a negative binomial distribution, and we can use DE-seq on the normalized count to calculate the differential expression. So you've done that for homework too, and you can also use lemma on the log level of the TPM, which, if you have more samples, actually this would work also very, very well. If you have a very few samples or few replicates, DE-seq is still considered a more sensitive method. And both DE-seq and lemma is because you may not have enough data to estimate a robust variance. It's using a hierarchical modeling for variance stabilization by borrowing information from all the other genes, especially giving weights to the genes that are expressed at similar level of your gene of interest. And that's how you gain power. And so with differential expression, we need to worry about multiple hypothesis testing correction because we are looking at every individual gene in the genome to see whether they are differentially expressed between your two conditions. And so we talk about two potential methods for multiple hypothesis testing correction. One is Bonferroni. This is to make sure that not even a single gene is predicted incorrectly, and it's very, very conservative. And so instead, we use more kind of a looser cutoff, which is false discovery rate, right? This basically says, out of all the genes I'm gonna predict, I just wanna make sure that my false positive is not over 5%. That's usually tolerable. Even I think 10% FDR, depending on the data, might be still okay. And then we talk about clustering. After you detect the differentially expressed genes, very often people eliminate those genes that are not differentially expressed at all and only look at the differential genes, and then you can cluster either the samples or you can cluster the genes. We talk about hierarchical clustering and how the different linkage, average linkage is most commonly used, but there is I'm not sure, it's like single linkage and the minimum and the maximum between the nodes. And we also talk about k-means, which have disjoint clusters, where a gene only belongs to one cluster, right? And you have to decide what is the k that you want to use, and we talk about the magic of 7. We also talk about consensus clustering. For example, if some samples are outliers or some genes are outliers, you want to just get the really tight ones together and you don't need to worry about the rest, then you can use this consensus clustering. You can also use consensus clustering to cluster samples if they are profiled with different platforms, right? How do we cluster samples if they're, the samples are profiled with DNA sequencing, RNA sequencing, microRNA sequencing, proteomics, and epigenetic profiling? We can still use consensus clustering to put them together. And then we talk about PCA. So PCA not only is a clustering approach, it's a dimension reduction approach. And the goal is to maximize the variations that are captured on the top principal components. And so you usually can visualize your result of many, many samples or in even just 2 dimension, even though each sample is represented by 20,000 genes. And from there, we talked about batch effect because very often you use clustering, either PCA or k-means or your hierarchical clustering, to detect whether there is batch effect. And if you only have 2 samples, you can use a combat on the log TPM. This can capture some nonlinear batch as well. You can also use lemma. This one consider mostly the linear model of the batches. And basically you are treating the batch as a treatment condition, and you're trying to see how much of that treatment condition is having on each gene as if it's a differential expression variable. And then we talk about DESeq2. So DESeq2, you can just look at whether the genes are differentially expressed. It will report the differentially expressed genes, but it would not give you a corrected expression data, whereas combat and lemma, after you run it, it will give you a new expression matrix with the batch effect removed. With DESeq2, you can still consider simple batches as another variable, but at the end, it will just give you the list of differential genes. I'm hoping that in the coming year, the developer DESeq2 can incorporate this feature so that not only it can detect the differential gene, but it will also give you a corrected expression matrix with the batch effect removed. Maybe they will do that. And then we didn't have this particular lecture just on machine learning because there are many good videos and whole courses on machine learning. We did talk about annotating the clusters after you get a gene expression cluster. How do you annotate the genes in those clusters? So there's a gene ontology, like a tool like David. You can also look at gene set enrichment. In gene set enrichment is you just look at the whole order of the differential genes, the Kamenov-Smirnov test, to see whether the genes annotated for certain gene ontology terms show a skew of the differential expression compared to the background, right? And then for classification or the machine learning, we very quickly discuss this unsupervised learning. You can just cluster the samples. You can do also MDS, right? The MDS is just based on the pairwise sample distance. You try to optimize that pairwise distance. And also PCA is to maximize the variance. And so, for example, if you use the correlation of the samples as the distance to do MDS, then PCA and MDS will actually give you the same result. And then in the single cell analysis, we also mentioned that using the k nearest neighbors to connect the cells that are similar, then you can use this modularity optimization to decide how many clusters there are in the single cells. That's kind of also unsupervised. It's not really for necessarily for learning purpose, but at least another kind of clustering approach. And then we talk about supervised machine learning. By the way, strictly k nearest neighbor is a supervised approach. Because you have to decide how big is your k to separate the samples. You will be using k nearest neighbor, logistic regression, random forest, and support vector machines in homework 3. So each of them, you will see that with this caret tool, that each is only one command line. You can do it. You just say, I want a 5-fold cross-validation, run KN, and run logistic regression, you run random forest, it will give you results. Yeah, so KNN, it works, it's simple, but it doesn't provide you with insight. It's just guilt by association. Logistic regression, you're trying to give weights to the different genes, trying to estimate, how to separate the two. And because there are so many variables, very often you have 20,000 genes and you only have a few, maybe 20, 50 samples to deal with. And so we need to deal with regularization. And regularization is in the cases when you have so many variables and also fewer genes, how can you robustly estimate those parameters? And by providing some penalty, so you want to create basically you want to not overfitting your data. So ridge regression still consider all the variables, but try to give them smaller coefficient. And lasso is trying to give a lot of zeros, so to make the coefficient really close to 0, and only keep the coefficients that are really important. And then elastic net is kind of a hybrid between ridge and lasso. And you will be doing this in the homework as well. Again, each of them is just a single command line. Then you will see how it performs. For random forest, we're building many, many trees, and each time you sample the data, you try to train the tree, and at the end, you again doing some majority learning, majority voting to decide how to make the final prediction. But then in the random forest in here, we use cross-validation to tune the parameters. And also a support vector machine is to maximize the separation margins between the two classes of data. And one way to have the good separation is to project the data into different other dimensions. So these are called different kernels. So you can try different kernels and see which one give you the best result. And so all of these, we need to make sure that the data is not overtrained, overfit, and therefore we need to do cross-validation. So for example, you can divide the data into five different sets. You train the data on 4 sets and test the data on the last that's left out. And you can try it many different times, right? So those are actually all now implemented with R packages that we can call on, which is really great. Okay, so then we have one lecture about single-cell RNA-seq, right? So this is kind of a recap. So we have smart-based and droplet-based single-cell techniques, right? We process the reads basically map the reads to the genome, get the matrix, and collapse the UMI for each gene on each cell. And then we QC the genes and QC the cells. We do dimension reduction of the big expression matrix. You can potentially remove the cell cycle effect. And then we can cluster the cells based on the modularity optimization. You can tune the resolution to decide, do I want 3 clusters or 10 different clusters? And then we can visualize those clusters with either t-SNE or UMAP, right? So remember, both actually focus more on the distance between the really points that are close to each other. The distant clusters, for sure, t-SNE, the distance is not scalable. With UMAP, the paper claimed that it's better, but there are people challenging this to say their data doesn't really support this claim. So it's probably slightly better, but still the distance close by are more reflective, but the distance far away is not as reflective. And then after that, we look at each cluster. You look at the genes that are in that cluster that are really differentially expressed for this cluster compared to all the other clusters. And then based on those marker genes, you can help annotate what cell type it is. You can also look at how each genes are expressed in different clusters. You can also visualize the marker gene expression in the different clusters. And then for differential expression analysis, if we are assuming a zero inflated negative binomial distribution, you can use the masked algorithm to detect the differential expression between the clusters, or in the same bigger cluster between 2 samples that maybe kind of overlap but have some slight differences. If we use non-parametric tests, we're not assuming that any distribution for the data, then we can use the Mann-Whitney U-test. And then we can use this canonical correlation analysis to correct the batches between samples by assuming that overall, most of the cells should overlap with each other between the 2 samples. And then there are many tools that are being developed, and we will use Surat for homework 3, but I think next year we'll see a lot more new tools and potentially better functions. Okay, so that's kind of an overview of all the material that we covered in the first